Hello, my name is Daniel Saban. I'm an associate professor at Duke University, and this tutorial will be focused on the immunology of age-related macular degeneration from the perspective of macrophage biology and pathophysiology. I would like to start by first thanking my colleagues, Kathy Bose-Rickman, Andrew Dick, and Chen Yu, for their thoughtful discussions and critical review of these slides. Now, this is a topical area of research, but it's also quite broad. It involves complement factors, as well as adaptive immune responses, and of course, other innate immune responses such as pattern recognition receptors, as cited here in the first set of bullet points. Our focus today, or in this tutorial, will be on macrophages, specifically of the neuroparenchyma. These, of course, include microglia. We now know these cells to be yolk sac-derived, long-lived, self-renewing, highly specialized, and locally maintained. And this is distinguished from monocyte-derived macrophages, which are adult bone marrow-derived and recruited in certain disease states. Now, early studies prior to when this distinction was established, or in human studies where this can't be resolved currently, we use the umbrella term macrophages in the current tutorial. The other thing I'll mention is that we quite often include information about mouse work in inherited retinal dystrophies, as these models are quite robust, and of course they allow us to study macrophages in very well-defined degenerative states. And in addition, both IRD and AMD both involve photoreceptor degeneration and outer retinal pathology, so there's quite a bit of homologies between these two systems. Our tutorial will be divided in 4 sections. Section 1 will be the significance of macrophages in AMD, 2, origins and function of parenchymal macrophages in the normal state, 3, distinguishing microglia in the degenerating retina, and 4, their functions in degeneration and their potential roles in AMD. Beside that, on the right, I have provided the anatomical layers of the retina, which you can refer to throughout the talk. And all the way in the corner, I've also provided a small icon of the retina, inner and outer, to show the particular orientation of the micrograph shown in subsequent slides. And that some of the images will be in reverse orientation, so that will be indicated in the right corner. So let us proceed to section 1, the significance of macrophages in AMD. Macrophage involvement in retinal degeneration was actually appreciated quite early. In fact, in the 1970s, here shown in the Rcs rat model of inherited retinal dystrophy, demonstrating the invasion of macrophages in the outer retinal layers where they're not normally residing. And one can see from these micrographs that the macrophages are actually enveloping pycnotic nuclei, suggesting indeed that these macrophages are phagocytosing dead and dying cells. Though not the focus of the current tutorial, which is macrophages in neuroparenchymal layers, it is nonetheless important to show this paper from 1986 in human postmortem tissues demonstrating the recruitment of macrophages in non-neuroparenchymal sites, such as drusen and the RPE. Following these studies came others. Here's one specifically in 2002, looking at human postmortem human eyes, either in retinitis pigmentosa, late onset retinal degeneration, as well as age macular degeneration. And in this specific figure here, we're showing from the paper the macula of an AMD subject with geographic atrophy and a scar. The staining is here done with RCA, which stains phytolectins, preferentially these are oligosaccharides expressed, specific oligos expressed on macrophage microglia, as well as endothelial cells and to some extent horizontal cells. But it's clear here with the staining, you can see again, invasion of macrophages in the outer retina, including the subretinal space. And in this inset in the middle in the white box, you can see an RCA positive macrophage that has rhodopsin particles inside, again suggesting phagocytosis of debris clearance. Several studies later on had validated those early findings with larger cohorts of post-mortem human eyes with AMD, including Ladd et al. In 2015, and some data shown in this review from 2017, specifically looking at the apical side of the RPE stained with IBA1 in conjunction with autofluorescence. One can see from these images that a healthy subject does not contain IBA1 positive cells in any significant number. And similarly, one can appreciate a same situation with small drusen. But this is in contrast to subjects with large drusen, where macrophages accumulate overlying on the apical side of the RPE. And finally, in geographic atrophy, specifically at the transitional zone, which is the edge of the atrophic lesion. Finally, we show this study here, which demonstrated the recruitment or invasion of macrophages again to the outer retina, specifically associated with subretinal deposits referred to in the current manuscript as reticular pseudodrusen, which are distinct from conventional drusen. To summarize section 1, it is clear that macrophages are involved in multiple steps along the progression of AMD, including subretinal deposits, large drusen, and of course in the final or late stages of geographic atrophy. Now, to understand their contribution in disease progression, it's important to now take a step back and look at the origins and functions of parenchymal macrophages in the normal adult retina, which is the subject of section 2. Now, recent breakthroughs have demonstrated now that the ontogenies of macrophages in different organs come from very distinct origins. So, for example, in the brain, microglia are derived from yolk sac erythromyeloid progenitors. In mice, this is as early as 7.25. And these are maintained throughout life without contribution from circulating monocytes. This is in comparison to other tissues, for example, the epidermis, lung, and liver, where you also have macrophages established prenatally, but they're derived from fetal liver monocytes, not yolk sac derived progenitors. And you also have tissues that have an open niche axis, no blood-brain barrier, with vessels highly vascularized, such as the heart, pancreas, gut, and dermis, where though they are initially seeded by prenatal macrophages, become outpopulated by bone marrow-derived macrophages throughout life. So what's very unique is these microglia, which are yolk sac derived. And this is really important to understand because their ontogenies shape, and we know this from various studies, shape their function. In collaboration with Florent Genou's lab at A-STAR, we recently demonstrated that adult microglia in the retina are indeed yolk sac derived, similar to the brain. To do this, pregnant dams, which were RUNX1 mercury-mer conditionally labeled, were pulsed with tamoxifen at E7.5, and the pups were allowed to age out to 8 weeks old, in which time the retinas were harvested and imaged for YFP colocalization of IBA1. And indeed, what we saw was that YFP microglia were found in both the inner and outer plexiform layers. indicating or effectively tracing these microglia back to the yolk sac. In that same paper, we subsequently showed that microglia are long-lived in the normal retina as well, which is in stark contrast to choroidal and posterior scleral macrophages, which were shown to be short-lived. To do this, we used adult mice that were CX3CR1 Cre-ER, conditionally labeled, by a pulse tamoxifen administration, and let these mice age 1 year following tamoxifen pulsing, at which time the retinas were harvested so that we can image and look for IBA1 colocalization of FGFP microglia, indicating that they're long-lived. And indeed, that's what we saw, both in the inner and outer plexiform layer. Over 85% of IBA1 positive cells were GFP positive, indicating that they are long-lived. And in contrast, as you see in the figures below, the macrophages that are IBA1 either in the sclera, posterior sclera, or the choroid were FGFP negative, indicating that they are short-lived. Now, what we also showed in this study, which was very interesting, is that though these microglia come from the same yolk sac origins, eventually, in the adult situation, they are maintained in different ways in the distinct compartments of the retina. For example, microglia in the inner plexiform layer are IL-34 dependent. This is a hematopoietin. In the case of the retina, it's expressed by retinal ganglion cells. So essentially, microglia in the inner plexiform layer are nurtured and maintained by the retinal ganglion cell layer. This is in direct contrast in the OPL, where these microglia are IL-34 independent. Now, even what's more fascinating, perhaps, is that microglia in both of these niches contribute to synaptic activity, as shown by us and also by Wang et al. In 2016 using depletion of microglia. One reason why this may be important is that these microglia may be the macrophages that are invading the outer retina, in which case they would be evacuating the synaptic layers and thereby potentially leading to synaptic deficits. Now, in this paper by Thanos in 1992 used the Rcs model, where the axon was cut and retrogradely labeled so that RGCs would then subsequently be labeled and eventually die. As they die, microglia in the inner layers of the retina would pick up or phagocytose these cells and thereby themselves become labeled. Now, what was observed is over the duration of the RCS progression, these microglia that were labeled initially in the inner layers began to accumulate after 30 so days in the outer retinal layers, suggesting that indeed microglia are the cells that are invading the outer retina. This is a rather simplistic view given the new knowledge that in degenerative states you get recruitment of monocyte-derived cells. And essentially these, in that previous model, potentially have been recruited in to take up that dye. Now here, this is work in the arrestin-1 knockout model, which is a light-induced degeneration model, with monocytes being labeled with CCR2-RFP at 24 hours post-induction of degeneration. These monocytes are arrested at the endothelial cell level in the vascular compartment, whereby at 48 hours you can see beautiful extravasation of these monocytes in the neuroretinal parenchyma. Then raising the question of which specific cell type might be invading the outer retina. Is it the microglia or the monocyte? That is the topic of the next section. However, distinguishing these two cell types is not a trivial matter. Here on the left side image shows that in CNS inflammatory states, you have microglia there, which are endogenous, prenatally established, long-lived, self-renewing cells. In contrast, you have monocytes recruited, again, in CNS inflammation, and these monocytes become or differentiate quickly into macrophages. And once they become macrophages, they are phenotypically indistinguishable from microglia. That is, their markers expressions very much overlap the microglia. Specifically, CCR2 gets downregulated, Ly6C, and then you get upregulation of macrophage markers such as F480 and IBA1, and the rest of this list. I'm just going to flash up a table which was published with the reference below, simply comparing and contrasting or showing the advantages and disadvantages of different mouse systems to study the microglia, which you can return back to at any given time. By far, the most robust system is the CX3CR1 Cre-ER mouse, which is established or developed by two different labs shown in the references below. The way this works, fractalkine receptor, which is expressed both by monocytes and microglia, upon tamoxifen pulsing, both populations become reporter positive. The floxed reporter stop codon allows for reporter expression for the lifetime of the cell. Now, in the case of monocytes, these are short-lived. So once they die, within 2 weeks, they're repopulated by hematopoietic stem cell precursors, and their progeny are no longer labeled. By stark contrast, the microglia, because they're long-lived, even in the absence of tamoxifen after pulsing, maintain the floxed allele. And even if these microglia, which self-renew, those daughter cells will also retain the floxed allele, thereby allowing you to distinguish, using this genetic scheme, microglia from the monocyte compartment. We were one of the first to actually use the CX3CR1 Cre-ER system as a lineage tracer to look at the spatial distinction or distributions of monocyte-derived cells versus microglia in degenerative states, which we used in the light toxicity model. So if you look at the top panel, you have YFP CX3CR1 cells, which include in normal, which are mostly microglia, as you can see labeled with TD tomato, both in the inner and outer plexiform layers. By contrast, under light toxicity settings, one can see YFP cells throughout the depths of the retina, including the very outer layers. However, when one looks at the TD tomato lineage traced microglia, they're only found within the subretinal space, whereas the remainder of the YFP cells are throughout the rest of the parenchyma. Likewise, using the same model with the same lineage tracing technique, by looking at the apical RPE in mice that were exposed to light toxicity, those lineage traced microglia adhere to the apical RPE, and most of them are indeed lineage traced microglia. And this is in stark contrast to the outer retina, which are YFP positive but tomato positive negative, indicating that these are monocyte derived recruits again. Using a slightly modified lineage tracing system, which conditionally labels or induces a diphtheria toxin receptor, which can be used for lineage tracing with immunolabeling or conditional depletion of microglia with DT or diphtheria toxin exposure. One can see in the top panel with light toxicity, there's YFP cells throughout the depth of the retina, but only in the subretinal space do we find lineage trace DTR positive cells. And interestingly, when one depletes these microglia in a conditional manner, as you can see in the bottom panel under, again, light toxicity settings, monocytes are spared, of course. Monocyte-derived cells recruited into the parenchyma readily. However, they still are unable to infiltrate into the subretinal space, suggesting that it's not a competition for space between microglia and monocyte-derived cells, but rather it's a restricted access that microglia specifically have to the subretinal space. A very similar finding was made using the same lineage tracing technique, but in a different model by a different group. This is the sodium iodide induced RPE injury model. This is a primary injury to the RPE and secondary influences on photoreceptors. Again, showing the TD tomato lineage traced microglia are the dominant macrophage cell type in the subretinal space, whereas in the parenchymal layers, specifically the ganglion cell, interplexiform, and OPL, are by and large tomato negative, CX3CR1 YFP positive, indicating a dominance of monocyte-derived macrophages. And finally, using the CX3CR1 lineage tracing genetic scheme onto the Rho P23H knock-in, which is a model of inherited retinal dystrophy, the same, yet again, trend could be shown. So in the top panel, just looking at IBA1 staining, in this degeneration model, you can see IBA1 accumulation in the subretinal space. Now, with the use of CX3CR1 lineage tracing with DTR conditional labeling, the cells or YFP cells that are on the apical side of the RPE are largely DTR positive, lineage tracing them, therefore, to endogenous microglia. And exposing these mice to diphtheria toxin in the bottom panel, you can see ablates these cells specifically. So to summarize, it is quite apparent from these multiple models, again, the light toxicity model, the sodium iodide injury model, as well as the inherited retinal dystrophy model, that endogenous microglia have dominant access to the subretinal space and the most outer retinal layers in the degeneration context. So with that information in hand, the question going forward becomes, what is the particular or specific function of these microglia invading the outer retinal layers, including the subretinal space, which is the topic of the next section. And knowing this, what are their potential implications in age macular degeneration? So to address this question, we recently applied single-cell RNA sequencing to identify the transcriptome of subretinal microglia using the light toxicity model. So it was done by FACS sorting microglia and macrophages from this model, as well as in the normal context, to find or identify different clusters of microglia and macrophage. With this information, we then applied pseudotime analysis, which is a bioinformatic platform, to help mine for the specific cluster of microglia representing the subretinal microglia, subretinal space microglia, which we then validated with differentially expressed gene markers of this population, one of which, shown here by in situ in the light toxicity model, is galactin or galectin-3. Once the transcriptomes were obtained, one could then identify, or using functional enrichment, gene enrichment analysis, upregulated and downregulated pathways. And what was interesting from these data was that many of the immune response pathways were downregulated in the subretinal microglia, suggesting that their functions are being performed in the absence of pro-inflammatory molecules or pathways. By contrast, what was upregulated are pathways involving lipid homeostasis as well as phagocytosis, such as in cell adhesion molecules involved again in cell-cell contact and phagocytosis, as well as antioxidant pathways potentially helping with reactive oxygen species. So from these data, we have a potential picture that these subretinal microglia may be protective rather than pathogenic. To address this question, what we did was to conditionally ablate microglia in the context of light degeneration. And in so doing, we can see clearly distinct phenotypes. So looking at the middle panel, in the presence of microglia, one can see in the fundus bright, distinct puncta suggestive of subretinal microglia. And in the OCT image, showing a degenerated retina. But this is in contrast to the depleted setting where microglia are absent, whereby in the fundus one finds a more confluent region of damage, again in the fundus. And corresponding to that in OCT, larger areas of hyperreflective events in the outer nuclear layer and segment regions, again indicating very distinct phenotypes in the presence versus the absence of microglia in the light toxicity setting. Now, looking histopathologically reveals more clues. In the presence of microglia, you can see a subretinal microglia there at 100x magnification, which is embedded in the apical RPE, delineated with an arrow. You can see clearly the inner and outer segments are gone in this particular lesion, and many of the ONL cells, much significant thinning. But this is in stark contrast with the depleted state, in the absence of microglia, where there's a massive debris zone shown by the red arrows, as well as a large accumulation of pycnotic nuclei in the outer nuclear layer. So in the absence of microglia, there seems to be a buildup of a very large buildup of debris, including photoreceptor debris, as well as photoreceptor soma. And lastly, when one looks at the apical RPE structure using phalloidin staining, a very clear differential picture here emerges. So in the context of light toxicity with microglia, there is some RPE dysmorphology and irregular staining, including potential phagocytic cups in the RPE. But this is in stark contrast with the depleted microglia setting, whereby one can see large areas that simply are not staining with phalloidin, potentially showing some atrophy of the RPE. In other areas where there's massive dysmorphology, as well as a number of phagocytic cups throughout the lesion. So it's clear that from these studies, there seems to be a protective role in that microglia migrating to the subretinal space where they undergo transcriptional reprogramming can function in a manner that restricts disease progression. So if that's the case, then how can we explain, if microglia are poised to protect the retina from degenerative disease, then how do we explain the development of AMD? Well, maybe that answer comes from the following concept, which is microglia immune checkpoint mechanisms. These are mechanisms by which maintain microglia homeostasis, allow them to function in the neuroparenchyma without collateral damage. And again, these processes have mainly been described, or at least discussed, in the context of development as well as in homeostasis, and to some extent in perturbed states of degeneration. Now, one such mechanism that we highlight here is CX3CR1 again. This is the fractalkine receptor. And the reason why this is highlighted here is because several studies in the retinal degeneration context with CX3CR1 knockouts have shown accelerated retinal degenerative disease progression in the absence of CX3CR1. So this suggests that this specific immune checkpoint may be key to maintaining microglial protective functions, and in the absence of such, may switch to a maladaptive state, which then is no longer able to protect or even causes pathology. So evidence from the latter, again, comes from the CX3CR1 deficient mice in acceleration of retinal degenerative disease progression. But also, interestingly, in subthreshold insults, such as advanced aging, as well as a light exposure, which normally pigmented mice do not respond to in terms of degeneration. In CX3CR1 knockout mice actually cause or lead to a level of photoreceptor degeneration. And a similar finding was made or linked to certain genetic risk factors such as ApoE4 and CFH, which of course are highly relevant in AMD. And in these settings, the absence of CX3CR1 leads to a microglial state which expresses high levels of CCL2, presumably helping recruit monocyte-derived cells, but not only into the parenchyma, but you see an abundance of monocyte-derived cells in the subretinal space, which have been thought or shown to contribute to photoreceptor damage, as opposed to protection or restriction of disease progression. So with this, one could propose a hypothesis in which adaptive responses actually maintain retinal physiology. And this is such even in perturbed states that are normally subthreshold, including advanced aging. And in the context of an inherited retinal degenerative disease, perhaps these adaptive responses play some role in restricting disease progression, although eventually overwhelmed by increased death of photoreceptors. And we know from CX3CR1 sufficient versus deficient studies that clearly CX3CR1 is one of the players in this adaptive response, but it's not restricted perhaps to just CX3CR1 alone. On the other side, you have a maladaptive response, and this could be in the absence of CX3CR1 or potentially in gene alterations of risk factors, or even compounded by non-genetic risk determinants such as weight, Western diet meaning, and smoking, as well as advanced aging. And in this maladaptive state, you can drive a perturbed state, which is normally subthreshold. And it can be that the progression of retinal degenerative disease is accelerated. And again, may involve these genetic variants and non-genetic determinants. From this framework, we can put forth the following hypothesis, shown in diagram form below. On the left axis, or y-axis, you see a protective factor index, such as lifestyle factors or genetic factors that actually contribute to protection. On the right-hand y-axis, you have an increased risk factor index, such as, again, genetic and lifestyle factors as discussed in the previous slide. And finally, you have on the bottom, the x-axis, which is aging. So what you can see, someone or an individual with high protective factor index can remain free of late AMD progression for the totality of their lives, whereas an individual with high risk factor index may succumb to late stage AMD quite early on in life. Now, potentially what contributes to one versus the other is the microglial response. Can a protective factor index contribute to an adaptive microglial response that in turn maintains normal aging? Whereas in an increased risk factor index, there is an increased maladaptive response and thereby contributing to disease onset and progression. And with that, I will close with the following slide, which demonstrates a similar thinking in the context of Alzheimer's disease, showing here how risk factors of Alzheimer's, including age, sex, and gene determinants, actually modulate the microglial response in A-beta plaques. And with that final slide, we will now close this tutorial and finally thank you for your attention.